please join me in welcoming Dr. Susan Wessler.
Thank you all. I'll tell you that the reason I'm most nervous about giving this talk is my daughter is sitting in the first row, and I don't think she's ever heard me give a talk before. So I'm delighted to be here today. And I don't know, for most of you, you probably don't know that the hardest talk that a scientist can give is, thank you very much, is to the general public. Because when we talk to our colleagues, we can sort of use all the language that we're used to, shorthand language. And for a talk like this, you really have to, much like what I have to do for my textbooks and for my undergraduate classes, deconstruct what you're doing to make it interesting, I think. And hopefully I've done that. So the topic, I think, is to me at least fascinating. I've worked on it all my life, all my science life. And I've divided it into several parts. And I'm going to show you that initially. The first one is I've divided it into the scientific problem. So this is really going to be a story about basic research and how I think in the 70s or 80s there was someone named William Proxmire who gave an award, the Golden Fleece Award. And I think certainly if he would have been around in the 40s, he would have given a Golden Fleece Award for this problem and for the public giving their tax dollars to solve something like this. And I want to show you why it's so important to work on problems like this. In this case, I'm introducing the problem, which is why are corn kernels spotted, by telling you a little background about the plant and a little bit about the scientist. I need to take this off. Then what I'm going to do is tell you about her solution to this problem of spotted corn kernels and how the solution eventually, after 4 decades, led to international recognition, and how something as seemingly trivial as spotted corn kernels has led to literally a revolution in biology. And finally, I'm going to end by telling you about how I'm attempting to bring my research to the undergraduate classroom and to high school classes so that they can sort of get the thrill of research rather than be bored with sifting through reams of material and textbooks. Because the reason we're scientists is we enjoy doing science, and that's really not what we're doing in the classroom. That's not what we're telling them about. So I've divided the problem into parts here. The first one is, again, stating the problem and pointing it out to you, and then telling you about the organism that was part of the solution, and that's maize, and about the incredible scientist who solved this problem, Barr McClintock. So to begin with, we'll talk about these odd phenotypes, spotted corn kernels. Again, talk about corn and talk about Dr. McClintock. So corn, many of you are familiar with corn, but I guess only in Thanksgiving do you really see it in its full glory, and that is where you see the diverse colors of corn. And this is Indian corn, and I know many of you are told that in organic stores or in food stores that blue corn is good for you. Well, it's really no different than yellow corn. There's nothing better for you. It is a lot prettier to look at, but there's really nothing better for you there. The difference is that blue corn has pigmented kernels, and this is just a digression here to explain to you why the kernels are pigment, and it's just basic, in a sense, biochemical genetics. And so the question is, why does maize have pigmented kernels? And it has pigmented kernels because in the alurone layer of the kernel, the pigment is produced. And pigment is produced because you have one compound, which I've just called A here, which is converted by enzyme 1 to B, which is converted by enzyme 2 to C. And finally, and this is in a normal kernel, everything's working. I've thrown a little more detail in here, just to show that this enzyme, each of these enzymes, is encoded by a gene. Okay, so we have the gene produces an RNA, the RNA produces a protein, and that protein in this case is an enzyme. And this enzyme, so there are 3 different genes for the 3 different enzymes. Too much detail, we don't need to talk about all that. The basic point is that to go from A to B, B to C, C to the pigment, we need 3 genes that are working. And they work when the pigment is blue or purple. Okay. Now, we're used to eating yellow corn. Yellow corn is mutant corn. Okay? And it's mutant because the plant cannot make the pigment in the alveolar layer. So I've just shown it here that what we have is we have a mutation in the gene. The gene cannot make the RNA, or it can, but it's not a good one. It can't make protein. And we have A, but A can't be converted into B. And that's all it is. And the reason I think they have us eat yellow corn is because it's blue food. I know there's a thing about George Carlin has a thing about blue food that, people don't like blue food. And I think it could get stuck in your teeth or whatever, and it's much better to have something that's kind of colorless. Okay, so now the problem is I've explained to you how we have pigmented kernels, and I've explained to you how we have mutant yellow kernels, but I haven't yet told you how we get spotted kernels. And I'm going to digress before I do that. Okay, so how does this happen? All right. What is the genetic basis for the spotted phenotype? That's the question that we're going to spend a bit of time trying to figure out. McClintock spent a lot of time figuring out. So that's it. So in order to do that, I want to introduce you first to the plant, because the plant is fascinating, maize, and to the scientist, who is equally fascinating. So we're all familiar with corn. When I was a graduate student, I worked with bacteria, with E. coli. And one of the nice things about switching as a postdoc to corn is that people understood what I was working with. I didn't have to say I'm working with germs. I could say I'm working with corn. So as you may know, corn is a major crop of the Americas. And up until recently, I was sort of proud to say I worked with corn. However, after reading The Omnivore's Dilemma by Michael Pollan and hearing about Archer-Daniel Midlands and what have you, it's sort of a working with the evil crop. The crop that is responsible for obesity in America and now is responsible for, the reason there's not enough cornmeal for people to make their native foods, basically. And it is a problem. But I love the plant. The plant is wonderful. And the plant is a domesticated organism. And it was domesticated from a plant, a wild grass called teosinte, and that's shown here. And for decades, it was not known what the wild progenitor of maize was. And the reason is pretty obvious. If you look at these two plants and you look at their seed case, you can see that there's virtually no resemblance between the two. They're completely different, yet they are members of the same species. Maize and teosinte can cross freely and produce fertile progeny. So this is an example of the teosinte ears. A lot of this work that I'm just going to mention briefly is done by John Dobley, who is a professor at the University of Wisconsin, who has really figured out what are the genes that were selected by early Americans and later on to turn this ear into the ear that we eat today, the plant that we work with today. So this is teosinte ear, and like most wild plants, the seed shatter, that is, they naturally come off of the ear so that the plant can support the next generation, so the seed can get away from the parent plant. That's one of the mutations that was selected for, that is so the seeds can be retained by the plant so that people can then come and harvest it. This is actually a trait, the shattering, that was selected for in most wild plants, in wheat and barley and other domesticated plants. This is to give you an idea of the scale of the domestication, how far domestication changed the morphology of this organism. This is the teosinte ear, as I said, and this is the ear of corn that was developed during domestication. Here you see our modern ear, and that was the product of a second process, which is called improvement. So there are probably only about 5 genes that were involved in converting this to this, and many, many more involved in the improvement from this to this ear. Okay. So corn was domesticated in Mexico. It is one of the few crops. There are a few crops that were domesticated in North America, in South America and North America. Most crops were not. Sunflower is another one, and there are a few more that were domesticated in the Americas. Jared Diamond's book, Gun, Germs, and Steel, is a wonderful story of domestication of not just plants, but of animals. So corn was not only was domesticated, but it is what facilitated the growth of early Mesoamerican civilizations, because people didn't have to go out and collect seed, and they didn't have to collect. They had time to develop culture. And so this is, as you see, the tremendous diversity that corn that we see with corn, the different shapes, the different morphologies. And it turns out that at the root of that is an incredibly dynamic genome. And that's where I'm going to sort of change course and tell you a little bit about that, beginning with the story of the solution to the spotted corn problem, and beginning with the person who solved that, who is Barbara McClintock. And as I said, Barbara McClintock, I don't know, some of you may have heard of her. Many of you probably haven't. She's considered one of the giants of genetics, with the other M's of Mendel and Morgan. She was a very unusual child. She was born in 1902 in Hartford, Connecticut. She was born to parents who were, her father was a medical doctor, and her mother was a musician, a piano teacher. And I only write this here because I am from the Bronx, and she moved to 1908 to Brooklyn, where she graduated later on from Erasmus High School in Brooklyn. As I said, she was an unusual child. Her parents kept her home from school, which was something that was allowed in those days, because they thought that she would learn more if she didn't go to school. She then wanted to go to college. She loved learning. She was a tomboy. And as you'll see in a later picture, her mother made her knickers, because girls couldn't buy knickers. They didn't sell knickers for girls. Girls could buy knickers, I guess. They weren't knickers for girls, and she liked to climb trees, so her mother would make her clothes for her. When it came time to go to college, her mother didn't want her to go to college, because she said it would make her unmarriageable, which she ended up being anyway. And her father prevailed, and he sent her. She went to Cornell, where she got a Bachelor of Science degree. She then, what I forgot to say, she wanted to go to graduate school, but she had a problem. She was interested in genetics, but the genetics department didn't accept women. So instead, she went to a department which accepted women, which was surprisingly plant breeding. And I say that now because it's kind of, genetics is a very accepting environment for women now, and plant breeding is a little less so. I'll put it that way, although it's getting better. So she got her BS in 1923. And then she joined the lab of one of the giants of genetics, R.A. Emerson. And there's a couple of buildings at Cornell named after Emerson. This is if you're a maze geneticist, which I am, this is one of our great pictures of the corn group, and it's taken in Ithaca in 1929, as you see. And this is for those of you who have been to Cornell, this building still exists. It's in the Cornell plantations. And it was sort of, when I was a graduate student at Cornell in the 1970s, she was just being re-recognized. And they started fixing up this building. And the only reason it still existed from 1929 is it really was neglected, and nobody wanted to tear it down and build another building, because it was an area of campus where nobody really, needed it for anything. So they were lucky, and they kept it. It was there. It's been refurbished, and you could visit it now with a little plaque on the door that talks about the corn group there. This is McClintock. You can see her in her knickers. These are the chute bags that are used, the tassel bags for pollinating corn. This is R.A. Emerson. This is Marcus Rhodes, who went on to be her dear friend and was responsible for getting her many of her jobs later in life. He went on to be head of biology at Indiana University. This is Charles Burnham, who was at the University of Minnesota. And this is George Beadle, who I think, being near Caltech here, many of you are familiar with. I'll talk a little bit more about George Beadle in a second. So this was a corn group. When Barbara joined the lab, the first thing she did within 1 week of joining the lab as a graduate student, she took on a project that people were having difficulty with, and that was trying to identify the different chromosomes of corn. She saw that they were going about it the wrong way. They were looking at the wrong material. They were looking at essentially female. They were looking at pollen and meiotic tissue, and she said you should be looking at root cells. So within a week, she was able to develop stains to stain the individual chromosomes, and she picked out the 10 chromosomes of maize. She was able to distinguish these. This is within her first month as a graduate student. She was probably the best cytogeneticist in the world, and this is sort of the old geneticists, people that would look at chromosomes and see all of the chromosomes, at the level that was possible then, that is a light microscope. So she was outstanding at what she did, but yet when she finished, she could not find an academic position. The reason being that women, it was very difficult for women to get positions in academia. They could work in their husband's labs or they could teach. And this didn't work for her. So Marcus Rhodes found her a job at the University of Missouri, where she started, but it didn't work out very well there. She was very unconventional. As I said, she was caught. She couldn't get into her office one day, so she scaled the side of the building to get in. She was not, she wasn't your average faculty member in many ways, but she also didn't want to teach. She really just wanted to do her research. And so Marcus Rhodes, again, found a position for her at what was the Carnegie Institute at Cold Spring Harbor. And that's where she went in the 1940s, and that's where she stayed for the rest of her life until 1992. And this is a picture of her in front of her microscope, and this is her in the cornfield. So there are stories of her. I guess the word is there's a legend about Barbara McClintock, that she was ignored, that she was unappreciated, and in fact she was highly respected by her colleagues. In 1944, at the age of 42, she was elected to the National Academy of Sciences. She was only the third woman elected to the Academy. And to give you an idea of the first 3 women elected to the Academy, 2 went on to win Nobel Prizes. So I think the bar was a little higher then for women. She then, in 1945, she was a wonderful colleague. She was elected president of the Genetic Society of America. So this was not somebody who we've heard in biographical accounts by some was ignored. She was highly, highly respected. So what I want to do is just quickly, before I dive into the problem of spotted corn kernels, come back to George Beadle, because I find him also to be a remarkable person. He essentially built the biology department at Caltech. He then went on, he was a superb administrator. He then went on to become the president of the University of Chicago. When he was at Caltech, he worked on Neuraspora crassa, where he did work that led to his Nobel Prize, which is the one gene, one enzyme hypothesis. But the whole time he really went back to his corn roots, so to speak. And even when he was at the University of Chicago, he would plant the fields in the back with corn. And he had a love for it, and when he was finished at the University of Chicago, he was the person who figured out that teosinte was the wild ancestor of corn. He went back after retirement, went to South America, did several crosses between teosinte and maize, and found essentially that there were 5 genes that were largely responsible, that Native Americans selected, that transformed this wild-grass into the sort of blockbuster crop that we have today. So out of this was a very illustrious group. Out of this group, we have 2 Nobel laureates in very different areas. So and this is a picture of Beadle and McClintock in 1979, on the 50th anniversary of this picture here. All right. So what I want to do now is go into the second part of the talk, which is her solution to the spotted corn problem, and introduce you to transposable elements. So that's the first part. Then I'm going to tell you about how this discovery of transposable elements really took a long time before it was generally appreciated by the scientific community, the brick wall. And then finally, her recognition, which came later in life. So again, we come back to the question, why are these corn kernels spotted? And just to remind you, what we've already established is that I've been able, hopefully, to show you, and most of you I'm sure know this, that we have pigmented kernels because this biosynthetic pathway is functioning. We have colorless or yellow kernels. I didn't have yellow here, sorry. Colorless, well, there's white corn also. Kernels because there's a mutation in the pathway, but the question then is how do we explain these spots? And through careful and years of detailed experiments, and this is a page from McClintock's notebook, she did thousands of crosses every year, and what she discovered was a genetic entity, a genetic element. And that's shown here, my sort of simplified version of her 30 years of work. And what she found was that there was a new class of mutation, which, unlike other mutations, was reversible. And it was due to the movement of what she called genetic elements. What she called them initially controlling elements. They were later called transposable elements, and I'm abbreviating them in this talk as TEs. So the difference between a quote-unquote normal mutation, which I know sounds ridiculous, but a typical mutation, let's say, and a mutation caused by a transposable element is that, remember I showed you before, if you have a mutation in a gene, like a deletion, or you have a base pair change, it's pretty permanent. The mutation does not change. But in contrast, she hypothesized that there were these genetic elements in the genome which can jump into a gene and cause them to lose their function, to be mutant. And then just as easily as they go in, they can jump out again. That they can excise from the gene. And I've shown that in this slide in the context of a spotted corn kernel. So we've seen already that what you have at the top is this mutant. So in this case, the mutant gene, which can't make a good RNA, which can't make a good protein, which can't make the enzyme, has a transposable element sitting right in the middle of it. All right? So the gene is not functional because there's a TE sitting right in the middle of the gene. That explains the white sectors that are in this kernel. However, what happens during the life of the kernel, as the kernel is developing, is that the transposable element, just as it went in, it could jump out again and go someplace else in the genome, anywhere else in the genome. When it does that, the expression of this gene is restored. We now can make a good RNA, a good protein, and we can complete the biosynthetic pathway and get spots of pigment. So that was her discovery, that there was this new genetic element that caused these types of mutations. Okay. So then she hit a brick wall, and that's what I'll just briefly describe, and that was her discovery of transposable elements were in the 1940s. She received a Nobel Prize for this discovery 4 decades later in 1983. It usually takes 10 or 20 years for somebody's work to be recognized as important enough to win a Nobel Prize. 40 years is a long time, even for things like this that take a long time. So it took really a long time for people. It's not that people didn't believe her, which is also another thing that's said, that, they didn't believe her, or they didn't realize the significance of transposable elements, meaning that she was working with maize. She was working with a domesticated plant. And people thought that what she was looking at was perhaps an isolated phenomena that was going on in maize, but maybe was not really important in other organisms. So, as I said, she ultimately won the Nobel Prize, and the reason for that, hopefully, is that since her discovery in the 1940s, scientists have discovered transposable elements in the genomes of virtually all organisms. Okay? In the 1950s, transposable elements were discovered in Drosophila, genetically discovered in Drosophila. In the 1960s, they were physically isolated from bacterial genomes. In the 1970s, the crowning achievement, I guess, they're discovered in the human genome. And remember, this is a Nobel Prize in medicine and physiology, so there usually has to be some relevance to human health. And so it was for that reason that in 1983, she won the Nobel Prize in medicine and physiology for a discovery, and I'll go into the discovery and the significance of it in a second. I think once you learn the kind of patterns, once you know that certain patterns are caused by transposable elements, you'll start seeing them everywhere you look. Most of what you see are not caused. Most patterns are just patterns. They're patterns of gene expression. Transposable elements cause patterns that look like this in a flower. This is actually a flower from a Sonoma vineyard. and you could see the sectors of pigmentation on the flower. What you'll also see are branches of the plant that have flowers that are either all white or all pink, all red. Okay? So I want to go into now her recognition, which, as you see, she's on a postage stamp fairly recently. So if I could sort of distill her discovery into one slide, it would be this one, that she had discovered a new component of chromosomes, a new resident of our genome. So before McClintock, people thought that the genome was essentially made up of genes, only genes. And I've sort of diagrammed that as these boxes being genes, and they're sort of like beads on a string. But McClintock's discovery made people realize that there were other things in the genome. That, in fact, after her discovery, we saw that there were these transposable elements, these pieces of DNA that could pick up and move into genes. Okay? So as I said, for this, she was the sole recipient of the Nobel Prize in 1983. Now, most people, you can win, I think, they will give out 3 prizes for a single discovery. It's unusual. It's not unprecedented, but it's unusual that somebody should get it alone. The fact that she got it alone really said that this was her discovery. This was her sole discovery. Not her sole discovery. She was the sole discoverer of transposable elements. One thing I did want to mention is I was fortunate enough to start my lab in 1983. And it is a wonderful thing to start your lab on a project that, all of a sudden is internationally recognized as important. So this is a picture of 50 years. This is also why I like working on corn, that the people who work on corn live a long time. So everyone here lived. Marcus Rhodes was almost 90. Charles Burnham was 92. George Beadle died younger. He had Alzheimer's. He was in his mid-80s. And Barbara, as you see here, lived until 1992. I had the incredible good fortune of knowing her the last 10 years of her life. And there's one incident that I recall. Well, there's 2 incidents, actually. One was I walked into her office after she had won her Nobel Prize at Cold Spring Harbor. And first of all, she hugged me and congratulated me because we had just published a paper on the cloning of one of her transposable elements. And I just thought it was so much like her to be so giving. But she had her Nobel Prize in lucite on her desk, and I held it up, and I'm looking at this thing, and she says, I almost gave it back. I was like, excuse me? I said, I almost gave it back, but I thought it was just, that would be a terrible thing for a woman to do. It would be very bad for women. So she said that. And the second thing I remember about her was when she met my father. So my father never graduated from college. He never went to college, and neither did my mother. He was picking me up. They lived in New York City, and I had stayed for a couple of days at Cold Spring Harbor. And he drove down. My father was a gruff kind of guy. He was an electrical contractor, somebody who was just fearless, I guess. And so he drove down in this big Lincoln Continental. We lived in a little tenement apartment, but he drove big cars. And so he drove down the street, and I said, Barbara, would you mind just talking to my father? And she goes, oh, of course, no problem. So they started talking, and they talked for a half hour, and I couldn't, for the life of me, figure out what they would be talking about for that period of time. And when I got back in the car, I got in the car and he took me back, I said, well, he just looked at me and he says, that is the smartest woman I've ever met in my life. He was just in total awe. And I said, what did you talk about? He said, we talked about our jobs. He said, because my father loved his job. I mean, he was a strange guy, but he loved his job. And that's what they talked about, how important it was to love your job. And that's what I loved about Barbara, was just that she was a very, people said she was difficult, but if she liked you, and she did like several people, she was an absolutely wonderful person. The sort of wonderful conversationalist, and just loved everything about life. She had this sparkle in her eye. All right, so let's move from there to I still really haven't explained to you what's so important about spotted corn kernels and what's so important about transposable elements, except that they're present in all organisms. All right, so to sort of convey the importance of this, I've divided this into 3 parts. First of all, what are they? How abundant are they? And sort of what good are they? Are they useful? All right. So this is the only really technical part, and I've tried to keep it as simple as possible, given that this is a after-dinner kind of talk. So this will only go on for about 3 slides or 4 slides. So transposable elements are pieces of DNA, and most of them, or a significant fraction of them, encode a single gene. And that gene is the protein that is responsible for making more copies of this element. They're a very simple system. There are 2 types of elements, and I'm going to show you how each of them move in just a few slides. These are the DNA transposons that McClintock worked on, and I'll tell you why they're called DNA transposons. And these are the RNA elements, or retro elements, that she didn't work on, that were worked on later. So this is, as I said, I'm going to be as simple as possible here. Transposable elements, I said, encode, in this case, a DNA transposon encodes 1 or more proteins, and sole responsibility is to catalyze their duplication and the insertion of the new copies elsewhere in the genome. And the way they work is, here is a transposable element sitting in a chromosome. It encodes a protein that I've shown here as a sphere. It's a transposase. This protein, what it does is it binds to the end of the element. So there's special sequences at the ends of the element that allow the element, these transposases catalyze the excision of the element from the DNA and the reinsertion of the element someplace else on another chromosome. And that's it. But if you're watching really closely, you'll see that there's no duplication involved. You're moving from one site to another. So for those of you that are of interest, I'll show how they can actually increase their copy number, and it's complicated, but it's just one slide. So we start here with 1 transposable element sitting in the middle of a chromosome. The chromosome it starts to replicate, it's copied. And when we have 2 copies here, one of the copies of the element will insert nearby, such that, if you follow this through, what you'll see is that we end up with 2 copies where we used to have 1 copy. And as I said, what these elements do is they make more copies of themselves. And I'll show you in a second, I'll tell you in a second, just how many more copies they make. This is the other type of element, which actually is a lot easier to explain. They're called retroelements. They're very much like retroviruses. This is a nucleus. Here is a retrotransposon. One type of retroelement. Like any gene, it is transcribed into an RNA, but this RNA is very different than most RNAs. It goes out into the cytoplasm, where it serves as a template for a process called reverse transcription. It's made into a DNA copy, which comes back into the nucleus, inserts someplace else. So on the next slide, I've simplified this so that what you have here is a retrotransposon. Here's the RNA copy. This element is basically like a printing press. It makes RNA. Every RNA has the ability, the potential, to be turned into a double-stranded DNA, and that can then insert elsewhere. So one element, it doesn't come out. It just makes copies of itself that go back into the genome. And this is how it increases its copy number. Okay? So I've told you what they are. I've told you how they make copies. Now I'm going to tell you how abundant they are, because this is the part that was really startling. So we're going to go to my favorite group of organisms, the cereal grasses. Here is corn, okay? And so I'm from the Bronx, as I said before, and I didn't know a whole lot about plants. I mean, the only plants we had in my neighborhood was the maple tree that was sticking in the front cement. And that was, fall came when they turned red, and that was it. So I didn't know a whole lot about plants. And the first time I really worked on plants was as a postdoc at the Carnegie Institution. So to me, you look at a maize, and for most people, I think, when you look at maize growing, when you look at sorghum, when you look at barley, wheat, or whatever, they look pretty much the same when they're growing. But what's really startling is now that we know a lot more about the genomes of these organisms, just how different they are at the genome level. So the rice genome, to start, rice has the smallest genome. Now, this seems like a big number, but it's actually a small number for genomes. It's 450 million base pairs, 450 million letters of the genome of rice. The genome of sorghum is 700 million base pairs. Okay? Genome of maize is 2,500 million base pairs. This is the size of the human genome. The human genome is more or less about 250 million base pairs. Okay? Barley, 5,000 million base pairs. Wheat, 20,000 million base pairs. And oat, even more. Now, for some of these plants, like wheat and oat, they also have, their genomes are larger because they're polyploid, and we're not going to go into that. But for the most part, these genomes are so big, not because, and I'll show you, I think, in this slide, that all of these organisms have essentially the same number of genes, about 30,000 genes. Okay? And this is pretty close to the number of genes in most complex multicellular organisms, whether they be plant or animal. About 30,000 genes. We don't know why it's a magic number. It's kind of a deceptive number also because a gene can be used for a lot of different proteins. But what differs with these organisms, remember, how can they have the same number of genes and this huge difference in genome size? It's the distance. In many cases, it's the distance between the genes. So if we look at rice, if these are 3 genes, the same 3 genes or similar 3 genes in maize would be further apart. What's between them are transposable elements. And if this was the barley genome, they'd be even further apart. So the genes are there, but they're embedded in this sea of transposable elements. And I think I use that in the next slide here. This is just an example from a colleague of mine, Alan Shulman. And here are 3 genes from barley. And each of these, they're drawn as little triangles, but you have to realize these are stuck. So this big sequence is stuck in to this place in the genome. This element is stuck into this one. This element is stuck into this one. So there's hundreds and hundreds of thousands of base pairs that are clusters of transposable elements, and somewhere buried in there are the genes. So this is what the genomes of many plants look like. And I think what I do with my introductory biology class is I ask them, usually, what fraction of our genome is derived from transposable elements? And they raise their hands. I won't make you do that. Say 1%, 5%, 10%, or 50%, over 50%. And the answer is over 50%. So over 50% of our genomes are derived from transposable elements. So, and the way I also try to make them sort of experience this is to say our genome is 2.5 billion base pairs, A, G, C, and T, on our 22. I'm a plant biologist, so I don't get the chromosomes right. Don't fault me. So this is equivalent to, and I do this with my introductory biology class because they have a textbook which is 1,000 pages long. So it's equivalent to about 1,000 textbooks of 1,000 pages each. That's if you have a letter, the same size font, the same letters. That would be the size of our genome in letters. No pictures, okay, no figures. All right? 20 to 40 of the 1,000 textbooks contain all of the information needed to encode all of our proteins. That's it. 500 of the 1,000 textbooks contain the sequences derived from transposable elements. Okay, that's fact, now that our genome is sequenced. And we are not unusual. So transposable elements comprise approximately 50%, as I said, of not just our genome, but the genome of other primates, chimp and ape, and other mammals. 75% of the maize genome derived from transposable elements, 85% of the barley genome, and to me, the most remarkable, 98% of the iris genome. Now, this is, you look at irises, they're beautiful, right? 98% of their genome is derived from transposable elements. 2% code for the proteins needed to make those beautiful flowers and the whole plant. So the question really is, how do organisms live with so much transposable elements? How do we live? I mean, this is when my students start to kind of scratch and start to get nervous. Oh my God, all of that stuff is in me. Well, the good news, I think, if I can do this, is that most of them are dead. Not the students, not the transposable elements are dead. They can't duplicate. They can't make more copies. They're inactive. They're like mutant genes. They have all sorts of mutations in them, deletions, what have you. They accumulate mutations, and the reason is because the cell normally accumulates when you replicate, when you copy DNA, there's a certain percentage that you're going to make a mistake. Those mistakes are selected out of populations because the organisms that have bad genes can't reproduce or die. Transposable elements, it doesn't really matter to the organism if they're alive or dead, so they just accumulate in our genomes. So that's the first thing. Most of them are dead. Okay? The other thing, which is something that my lab has been very interested in, is that transposable elements have evolved strategies to insert into safe havens in the genome. They have evolved ways to go places where the organism will not sustain, some sort of serious, won't die. Let's put it that way. All right. The other thing, which I'm not going to have time to talk about, is that the host regulates transposon movement. And this is actually really interesting. And this would be a whole talk, because it turns out that some of you have heard about epigenetics. It's sort of the silent genome. It's like the dark matter of the genome. It's the regulation that's sort of, that's not encoded, but it's the responsibility of, whether a chromosome is condensed or open, and whether genes can be turned on or off. Well, it turns out that probably that mechanism has evolved to keep transposable elements under control. I'm not going to talk about that further. But what I want to show you is this would be a typical human gene, and I'm not going to go through not basic biology, but college level biology here to tell you that our genes are made up, they're encoded in pieces, exons and introns between them, and that the introns are the parts that are edited out of the RNA. But the point I want to make is that this would be a typical human gene, and a typical human gene can have hundreds of transposable elements in it, in the gene. And generally, they are in the introns, where they are removed from the mRNA. So these elements now, in this case, if you can imagine that transposable elements can insert anywhere in a gene, but those that insert into the exon, that would be a detrimental mutation, and an organism that has that mutation would either die or be weeded out of the population. So what's left are sort of the neutral mutations, the ones that are in places that don't harm the organism. One of those places are in the introns. This is to give you an idea of how our genome is, we've said, is like an ecosystem. So here I showed you this before, genes. Here's a part of the chromosome where you have these gene islands with transposable elements between them. And so the elements actually many of them have a preference to go into each other. So they keep making more and more copies, and those copies insert into each other. So you build up these huge clusters, and it literally drives the genes further and further apart. Now, then we have other kind of elements that, as I said before, go into the introns. So these are highly evolved relationships where transposable elements make more copies, and the ones that are able to duplicate while not harming the host will be successful. Okay. And those are spliced out. So now the question that we're still approaching, there are some answers, but definitely it is an open question, are transposable elements useful? We know some are useful, but McClintock actually was the first to propose a function for them, or a role for them. And she said that they were tools of evolutionary change. So what she said is that transposable elements usually do not move around, that they're quiescent in effect, that there are conditions that can, and when I say stress, I don't mean, driving in rush hour traffic. I mean dramatic temperature changes, where a population is now subjected to maybe a 10-degree increase in temperature. It's one of the experiments that many of us are thinking about planning to do, is looking at different populations of organisms and seeing the impact of global warming, for example, if it is in fact activating transposable elements in organisms. Okay? So what may be bad for many people may be a golden age for transposon biologists. Right? So what she said is that their movement increases the mutation frequency. Okay? Now, most mutations that are caused by transposable elements are usually neutral, have no effect, or they're harmful. However, there's a rare transposable-induced mutation that could be adaptive, that could be good. So she's saying that this is an organism that's sort of rolling the dice. Not an organism, a population that's rolling the dice. It's saying, okay, times are bad, and we have to really shake things up. And so how do we shake things up? And again, this is very I'm not going to go into great detail, except to say that transposable elements, when they insert into genes, and I've just shown this, you can think of this as the exons in a gene or whatever. This is one gene. If a transposable element sits itself down next to the gene, it could alter the way this gene is expressed. So you could have a gene, for example, in a plant that was expressed only in the leaves. And this transposable element being in the control region, the promoter region of the gene, the place that determines whether it's going to be on or off, can change the signals, and this gene maybe now will be expressed in both the leaves and the roots. And it could be possible that the presence of the product of this gene in the roots is advantageous under certain circumstances. So it's not knocking out the gene, it's making the gene product available at other times. That's one thing it can do. This is kind of complicated, and I'm just going to show you really quickly. I've shown you exons and introns. A transposable element can actually change the way the pieces of a gene are put together so that normally the intron, which is shown here, is removed, giving us this normal RNA. But the presence of a transposon could also cause a piece of the element to be present, and have this little piece then could be then coded into the protein and change or alter the function of the protein. The interesting thing here is that we're still making the correct gene product, but the organism is diversifying by also now making this second gene product, which may or may not be of use. The third way, this is just a textbook figure to show that we're most of us, or many of us, are familiar with gross changes that can occur in chromosomes. So what I said is TEs can promote not just these little changes in chromosomes in genes, but major rearrangements. So here we have a normal chromosome, and that chromosome can do a lot of things. We can have a piece that could be deleted. That's usually not a good thing. We could have a piece that moves from one place to another. And we can have a piece, this is the most significant one that I want to talk about. We can duplicate information. And this, in fact, is something that transposable elements are very good at, not just duplicating themselves, but duplicating sequences around them. And it turns out that what we're finding out from sequencing genomes is that this is a real driving force in evolution, that if you have a region of the genome and you copy it, and there are genes in that region, those genes now are free. They're not constrained anymore, and they can be free to adapt. They can be free to diversify and possibly allow the population, the species, to adapt to another environment. So the idea, and that's been shown, so for example, if you look at the rat genome and you compare it to the human genome, one of the major differences are the number of sensory receptors that are in the rat genome. Rats have many, many more sensory receptors, smell receptors, than we do. And they use them because their eyesight isn't very good, but they can smell their environment. And this is due to the duplication of these receptor genes, allowing the rat to adapt to different environments. And what I've shown here, and this is just really, all I'm saying here is that this is a fairly recent summary slide from a wonderful researcher at the University of Washington, Evan Eichler. These are the 23 chromosomes of humans, and since a genome's been sequenced, Evan has found that about 5 to 6% of the human genome are regions that have been duplicated. So all of the red regions you see here are regions that have been from somewhere else that have been copied and stuck someplace else in the genome. So this is a huge fraction of our genetic material that is sort of possibly free to evolve into another function. Now, the vast majority, when something gets duplicated, it's just a random event. It happens. Any part of our genomes can be duplicated. Okay? The ones that are retained are the ones that at some future time may prove useful to the species. All right, now you can rest. So basically I wrote this in a grant application once, and that is that transposable elements shake up an otherwise conservative genome in ways that we are really just beginning to understand. They're sort of like I had a cat, an old cat once, and I brought in a little kitten, and it keeps the cat on guard. I mean, the way also I like to think about it is organisms have to deal with lots of pathogens, one thing, viruses, that they've never seen before. And one of the things that transposable elements have done is their movement, their insertion into sites is very similar to a virus inserting in the genome. And one of the reasons that organisms can protect themselves from many viruses is because they evolve strategies to protect themselves from transposable elements. So transposable elements are sort of like the system within that sort of keeps all life on their toes. And you might say, well, what happens? What happens if they just keep amplifying? Well, the organism dies, and most species are extinct. So the one thing about life is that living things are that they have survived. They have figured out how to deal with their elements. The ones that haven't, we don't know about. All right. So what I want to do is, as I said, end on a lighter note and tell you about a program that I've been involved in for the last year or two, which is, I think, a wonderful, wonderful program. And it's sponsored by Howard Hughes, and some of you may be aware of it. There's someone in the audience who is part of this program, I see, at Irvine. And that is a Howard Hughes HHMI program, and it's a professor's program. So the idea really is that there has never been a more exciting time to do science. Certainly biological research has never been a more exciting time to do science. But yet we are managing to sort of bore our students to death with huge textbooks and a huge just huge amounts of facts that we're dumping on them. And what we're, the reason that we got into science, the reason we're doing science, they really get what's I'm trying to say? They never experience it, until it's too late in many cases. So what Howard Hughes initiated 6 years ago was a program called HHMI Professors. And the idea was that in a variety of venues and a variety of mechanisms is to bring the excitement of research to undergraduates and to high school students. And they do this in the way Howard Hughes does it by putting a lot of money into something. In this case, it's $1 million for 4 years to develop a variety of research teaching programs. And my program, which I'll just briefly say, is really to do what I do in the lab, create a lab environment for students. So my provost gave me $250,000, I call it my second startup, to design a lab that was modeled, a classroom that was modeled after my research lab, which is both computational and experimental, for undergraduate classrooms, undergraduate laboratories. Now, there's lots of classrooms that do that, lots of classes that do that. The difference here is that I was able to focus on what we do in my lab, that are transposable elements in the genomes of high organisms. So essentially, the reason they were also interested is we're using transposable elements, their experience with transposable elements, to teach them about evolutionary concepts. And many of you may know that my state and other states, not just in the South, have a problem with teaching evolution to students. My state put a sticker on introductory textbooks saying that evolution is just a theory, just like any other theory. And so I think putting okay, enough said. I won't go into it anymore. All right. The other thing is to collaborate with local high schools in my area, which is something that's just getting off the ground now. But I also have the privilege of collaborating with a wonderful school in Los Angeles, High Tech High LA, to bring a variety of modules to their biology program. So why focus on transposable elements? They're simple. They're relatively simple. They're, as you see, incredibly abundant. They evolve rapidly. They promote genome change. And most importantly, they're largely ignored. And the reason that's important is that students then could actually be involved in discovery. So they can do things. They can be doing things for the first time anyone has ever done it. They can look at a genome. With my $1 million or $250,000 a year, using new technology, I can spend $10,000 to have a draft sequence done of a particular plant. The sequences can come back, and the students in one semester can analyze the sequences and be the first ones ever, to look at the DNA of a particular organism. And this, it turns out, is very important for students, and I think rightfully so. This is a class from High Tech High. One of the really neat things is that they started a module working on maize, and about 80% of the students in the school were Hispanic. And maize also was something very important to them because it was something that was discovered in Central America, and it's an important part of their culture. So they really took to this module. So I wanted to end by just saying that I hope I have shown you that focusing on a seemingly esoteric problem, having a particularly brilliant human being focus on an esoteric problem, can really change the way all of us do science and lead to things. In this case, that 50% of our genome and most of the DNA on this planet, I would say that, most of the DNA in higher organisms on this planet is derived from an entity that we didn't even know existed 50, 60 years ago, transposable elements. So I think and I wanted to end actually with a quote from McClintock's Nobel lecture in 1984, where she talks about the genome, and I really think she's probably one of our first, as we say, genomicists, where she talks about, I'll let you read it here, I don't have to read it. There's another page here. Again, this was somebody who was unbelievably ahead of their time, and it really took 40 years for even the scientific world to catch up with her, and we're still trying to catch up with her. As I said in the next slide, that we know about the components of the genome that could be available for restructuring, but we know nothing, and still to this day we know very, very little about how the cell sort of mobilizes its elements, or how the elements are mobilized, creating the genetic diversity that we see, a lot of the genetic diversity that we see. So I think I will stop there and be happy to take questions or